Good morning, everyone. I'm Dr. Margaret Stuber, a professor of child and adolescent psychiatry at UCLA, and also the director of the residency program at the West LA VA, or Greater Los Angeles VA. And I am delighted to introduce my colleague at both places, Dr. Erika Nurmi. I'm going to briefly go over her CV just so that you see why I'm so impressed with her, but briefly so that she gets to talk. Dr. Nurmi got her MD PhD at Vanderbilt in neuroscience. She is an associate professor in residence at UCLA, is the division chief for anxiety at the VA, and is in charge of two child programs in the community also dealing with anxiety. She's a journal editor for molecular neuroscience and for iScience. I didn't even know there was that yet, but of course there is. She has been very involved with medical student education, psychiatric resident education, and child fellows, particularly mentoring people in research. She's gotten teaching awards at the VA for the last 4 years in a row, on top of many teaching awards over the years, including the Semel Research Mentor Award. She has 53 research publications with 4 under review. 9 of these are first author publications. She's PI on 1 current grant and co-investigator on another, and has been, as far as I could tell, continuously grant funded since 2009. We are delighted to have you with us, Dr. Nurmi, to share some of your most recent work. Thank you.
Thanks, Margie. Very happy to have Margie introduce me. She's one of my favorite and most important mentors over the years. So that is special for me. Anyway, I will share my slides for you guys. I did leave the chat open. You're supposed to ask questions in the Q&A, but if you have, there's a few interactive things where you could put things in the chat, and I'll encourage you to overcome your social anxiety and put things in the chat when I ask. It's a good exposure because, you know, I have to be wearing that hat too. All right, so today I'm talking about diagnosis and treatment of mental illness as seen through the lens of genetics, thinking about where we are today and where we are going tomorrow. There's really been a lot of exciting advances in genetics, and I don't know how much they trickle down, but they are going to be part of our treatment in the years to come. So just a few disclosures. I don't take money for my advisory board activities, but I do have some. I also have some paid clinical trials I'm working on. I guess that CV was a little old. And then also NIH funding. But our objectives for today, we want to understand how genetic advances have begun to reveal the biology of psychiatric disorders, learn the implications of recent lessons in psychiatric genetics, and consider the ways that future genetic discovery will change how we practice psychiatry. And as we go through, we'll go back to this outline to kind of orient you on where we are. But I want to talk about how we finally started finding genes for psychiatric disorders, because for so long, you know, we had the promise of the Human Genome Project, which was finished, oh gosh, almost 20 years ago now. And what have we really gotten out of that, and why that's taken a long time, and how those fruits are finally coming to bear. What we've learned about psychiatry and recent genetic findings, and then where we are and where we need to go for genetically informed psychiatry, which I know we're all super excited about. So starting off, how we finally started finding genes for psychiatric disorders. The disorders we treat are very much genetic. So there's a couple of measures we use to estimate that. Sibling recurrence risk means we compare if a first child has the disorder, what's the likelihood the second child will also have the disorder versus the population risk? Because that tells us if that second one has it, and that's much more than the population risk, there must be a lot of genetics there. We also use heterability, which is calculated by comparing how much genetic information two relatives share versus how much they share phenotypes. And you can see that our common disorders are very much influenced by genetic factors. And the other thing to notice is that there's still room for us to intervene, right? Because environment, which we can alter, is a big part as well. So the problem was we completely underestimated the complexity of psychiatric genetics. And for a decade after the human genome sequence was available, we saw repeated GWAS failures, genome-wide association studies, and no significant hits because our sample sizes were woefully too small. There was a period I was going to conferences and every presentation was, oh, and we didn't get any hits. We didn't get any hits. And it was very depressing and very sad. And people were telling me at the time, and I was just really getting, I think, into residency at that point, get out of psychiatric genetics. It's a dead end. It's going to ruin your career, right? But thankfully, I didn't do that because now it's quite exciting. Um, the other thing I wanted to point out is that we're going to see some of these slides that look like this, and this is the Manhattan plot. I don't know if anyone can guess why it's called a Manhattan plot. I'll let you think about that and I'll reveal the answer later if you don't know. But here we map all the chromosomes we have, all the spaces in our genome, and all the variants we test when we do a genome-wide association. And if the variant is highly associated, it gets a high score. And so we get these dots up here that are much more interesting than the dots down there. So I'll come back to that. The thing that really changed for us, where we started actually being able to get genes for psychiatric disorders, were really about scaling up and big data. And so the timing worked well because big data became a thing in every field. But three advances made it possible to achieve adequate sample sizes. We started having these population registries. Australia does this, New Zealand, Norway, UK, and not so good in the US, but a lot of the other countries do it where the whole population gets put into a registry with all their electronic medical records and their genetics as well. And then we also have these disorder consortia that were created. Many of them, the largest is led by, in the US, by the Psychiatric Genomics Consortium. And they just are pooling everyone's samples to create huge sample sizes. So we really had to learn to share in science. So my child psychiatry comes back to me often, and that's a way where we really had to get researchers to share and provide these large numbers of samples that we needed to be able to have enough power to do these huge scale studies. Technology improvements also facilitated rapid, high throughput, affordable genotyping. So it used to cost a fortune just to do, pretty small experiments. And now we can do for $50 or less the whole genome markers through the whole genome. So that made a huge difference. And then computational methods were integrated to manage and analyze all the data we generated and new statistical approaches that could really ask interesting questions. This is an example of a microarray, the technology that made this high throughput genotyping possible. And so you can see that little tiny chip that's in the person's hand is one of the first arrays, the Affymetrix chip, and they look a little different today, but same idea. And one person's DNA goes on that chip, and then if you blow up the little cells on that chip, you can see that there's little dots that are all the different markers across the genome that get tested. And a laser is able to see the identity of the base pair that's at that location. And so you can do the whole genome in one blast, not very much money, for less than $50. So that was really important. The big population data has also been very important because we don't want to just study disease. We want to study population variation. And there is disease in here and there's healthy people in here. And so you're able to ask a lot of interesting questions of that. It provides a more comprehensive landscape on influences on broad phenotypes, not just disorders. So you can look at all kinds of different cognitive, personality, behavioral traits, and you get lots of power because you have these huge sample sizes. But the phenotype data is not very detailed or reliable because it's really self-report or EMR mining. Um, And so sometimes you can really make some false conclusions because this is not a well-controlled study, right? You're just kind of grabbing information, and there could be other factors in there that are leading to false conclusions. And if I had more time, I'd give you some cool examples of that, maybe next talk. But one of the leaders here that makes all their data public is the UK Biobank. They've been amazing. And really just collecting UK citizens and doing all kinds of testing. They have their EMR, they have all of this extra data they get. They do all these self-report measures. They even do computer cognitive measures where they're like doing a trails or some kind of attentional test. And they do imaging even. And so we were able to see some really interesting long COVID results with that. But the UK Biobank has been a great resource, and it's open to all investigators to take that data and analyze it, which is amazing. Back on the home front, the PGC, the Psychiatric Genomics Consortium, has really led the way in collecting these huge sample sizes. You can see in the table, this is a little bit old, but only a few years, where you have all the disorders and the number of cases and controls that have been collected for those disorders. Overall, more than 500,000 cases with one of the PGC psychiatric disorders have been collected, and they've had genome-wide significant findings at 445 loci, which is amazing. I don't know if you guys have really appreciated that we've really had this kind of success in psychiatry with finding genes. Loci is not genes, of course, it's a piece of the chromosome, but the next step is figuring out the genes, and I'll show you some examples of that. But interesting thing that emerged is a lot of these genes are shared across disorders. And that may not surprise us that much as psychiatrists, because how many patients do you have that don't have comorbidity? It makes a lot of sense that genes that would be involved in resilience or impulsivity might really affect a lot of different disorders, right? And the PGC has said they'll have greater than 2 million cases by 2025. So this continues to grow and give us more power to find more and more data. The first GWAS victory really emerged in 2014 from the schizophrenia GWAS consortium in the PGC. And this figure was shown like a gabillion times, but it really does illustrate a different kind of Manhattan skyline where you see this very tall building. So there's my Manhattan plots are named because they look like skyscrapers when you have hits. And so these guys did almost 40,000 cases and over 100,000 controls and saw 108 genome-wide loci, 108 pieces of the genome that strongly were associated with risk of schizophrenia. And I usually ask the students when I give this talk, what's that number 6 really tall skyscraper? And you might remember, if you're a genetics dork like me, that chromosome 6 is the MHC locus and the immune genes, and that's the complement protein that was shown that really started driving a lot of interest in the idea that neuroinflammation is involved in schizophrenia. Now, today, remember that was quite a few years ago. Now in 2022, the PGC schizophrenia GWAS is still leading the pack. Might be some offended by that statement, but that's what I think. And they've converted a lot of the loci, the places on the genome, to actual genes and function now, which is really the next step. A lot of the times you may not have heard about some of this research because if you just find loci, well, okay, what do we do with that, right? If we don't actually have the genes, it's not telling us much. So they've been able to start fine mapping to actual genes that are being that are driving those signals. And what they're finding is related to function. So they're often expressed in excitatory and inhibitory neurons. We're really starting to appreciate in neuroscience that the excitation inhibition balance is really important. And when that gets messed up, that can lead to psychiatric disorders, a number of different ones. OCD is thought to be one of these as well. And they also found that the majority of these genes are involved in synaptic functioning. So it's a little bit of a confusing figure, I think, but what you're looking at is that the brighter red something is, the more genes localized to that area. And this is kind of a map of the synapse. And so you can see pre-synapse, post-synapse, all these genes are right there at the synapse, right? When you look in the neuron at where they're expressed, much less in these other areas, everything's really centralized to the synapse. And you can see what those individual genes are over here. And so that makes a lot of sense. I think that in schizophrenia, you might have slightly imperfect, unideal, unideal synapses forming. And so this is where you start to connect the genetics with understanding the function and understanding what's going wrong in these disorders. Okay, now we're going to move on to what we've learned, some recent findings that we can get excited about. So anorexia is a one that was slower to get going, just like with OCD and Tourette that I'm in. But they had their first victory GWAS reported in 2019. They saw 17,000 patients with anorexia and 55,000 controls, and they found 17 significant loci. But what was really cool about it is that there was a striking face validity of the genes because they impacted genes that were both psychiatric and metabolic. And I have to have a shout out here to Mark D'Antonio, our beloved Mark D'Antonio, who's going to have the inaugural lecture dedicated to him. I forget what month, in the next few months. And Mark was an amazing force of nature who we all cherished, but he was also brilliant, man, because when I was a resident rotating in my second year on the inpatient child service, Mark found out that I was interested in genetics, and he took me aside and he said, listen, when they find the genes for anorexia, they're going to be both psychiatric and metabolic, because he said you can't just have the obsessive need to control your weight, but you also have to have the ability to do it. So you have to like be able to lose a lot of weight, and you have to be able to restrict, and not everybody can do that, which boy do I know. I'm just always like, how do the anorexics do it? I like have a terrible headache if I don't eat. Oops. And so actually I got to tell Mark that he was right. And we were, I think, one of the child graduations, and I went and said, Mark, you told me in 2006 when I was a resident that this was going to happen. And the genes for anorexia, some have been found, and you were right. They're both psychiatric and metabolic. And he was like, yeah, duh. You know, I was like, oh, that is so Mark. He wasn't even like proud that he got it right. He was just like, yeah, of course, right? It's like funny. But I was impressed. I'm still impressed. Another really cool thing that they saw in the anorexia data is that there really is an incredibly strong overlap with OCD, which probably doesn't surprise any of us child psychiatrists because there is a lot of comorbidity and also a lot that feels very similar, right? And so when they did a cluster analysis, you guys might know about cluster analyses. You can do these with all kinds of factors, but genetics is one of them. You actually see that when you map the anorexia risk genes that are in that GWAS, they align very well with OCD and the compulsive disorders like Tourette. So this with cluster analysis I thought was very interesting because they have very strong psychotic disorder, schizophrenia, bipolar link is very strong. And then the internalizing disorders, major depression and anxiety makes a lot of sense. Now, this one's curious. And ADHD and PTSD keep coming up linked together in different studies. So that looks real. And I wonder to what extent, I have to look this up, but the PTSD, I wonder if it's all coming from veterans in those studies and it's all men, because it makes a lot of sense to me that that link would occur because there's huge ADHD in the military, which sure, if you're struggling for structure and you have a hard time with the school system, you get out of school and you're like, oh, the military makes sense, right? So we have huge ADHD in the veteran population, and then we traumatize the heck out of them and they have PTSD. You could also say there's definitely genetic factors that are involved in risk for trauma, which might be about impulsivity. And so not just risk for poorly recovering from trauma, but also separate genes that predispose to having trauma. So I really would be interested to see if this was true for women who have trauma, because this may be more of a male effect. Of course, autism is also loading on this factor with the neurodevelopmental ADHD. Alcoholism is a weird one. I think this is just, it gets grouped here because this is the strongest thing for alcoholism, but it's kind of linked almost equally to all of the factors. So that may be a little different. Anyway, the large GUS that we have in these PGC consortia can tell us a bunch of different things that that are really providing us some interesting big picture ideas. So you can look at genetic correlations between disorders, and you can also look at shared genetic risk between phenotypes of interest. So we're able to really start making comparisons, like how are these things genetically related? So I'll give you some examples. This was a great paper. That looked at all the PGC disorders and saw what are the connections between them, what's the overlap, and what you can see. You don't want to look at the diagonal because that's the disorder with itself, but you want to look at the blobs, the dark blobs. So you have one here, bipolar schizophrenia, you have one here, OCD and anorexia, and you have one here, that's the PTSD, major depression, anxiety. And here again is your PTSD and ADHD. I thought that was really cool. But what I thought was even cooler is that when you look at neurologic disorders, they don't overlap with each other. There's something special about psych disorders that they do overlap. And there's very little overlap between neurologic disorders and psych disorders. For a long time, people joked, well, things are psychiatry if we don't know anything about them. But once we have biology, they're neurology, right? But that's not true. There's actually something different about psychiatric disorders. So it really, it makes sense. And the neurologic disorders that do overlap are things like migraine, which of course has a lot of functional or psychiatric input in it. So that also makes sense, right? And we're really starting to think about, the way that we describe things with the DSM is useful sometimes, but maybe not really a good reflection of biology, right? Because what we really have is population spectra of traits. And when you're at the high end of that spectrum where you have a lot of genetic loading for something, now you're impaired and you're distressed and you qualify for a DSM diagnosis. So when we're looking at the population, we see the spectrum of, say, neuroticism, because I study anxiety, and some people have low neuroticism and some people have high neuroticism, not speaking of anyone specifically like myself. Uh, but when you get to this certain point where you're impaired, now we're calling it a categorical diagnosis, but that's artificial, right? And so we really have to think about the underlying structure when we talk about genetic disorders. So when we study the genetic overlap between things, we often use a polygenic risk score. You guys have probably heard that term come up. It sounds much easier than it sounds. The idea is even though we know in all of these variants that are more strongly associated than these variants, even though we know a lot of these are false hits, that they're just associated by chance, we know that the signal is mostly up here and the noise is mostly down here. Now there's some noise here, but most of the signal is captured with those. So if we take all of the variants that are associated at a certain significance level, which is kind of debatable, and you can use fancy statistics to figure it out. If you take those collection of variants that have a higher contribution, and then you look at those same variants in another disorder and see, do these variants that predict risk for schizophrenia also predict risk for bipolar, also predict risk for depression, whatever, right? That's all they're doing. They're taking highly associated variants in a collection and looking at them in different disorders to see if they share those same risk variants. Hopefully that makes sense. I hate webinars because I can't see if any of you are understanding or anything like that. But that's really the key. And so here's an example, ADHD, they found 12 significant loci in their most recent GWAS. And what they saw was super interesting in several areas. So one, ADHD diagnosis overlapped with population-wide cognitive traits. So it's just like we said, it's a spectrum of sort of impulsivity and attention, difficulty focusing, cognitive control issues that we see in the population. And the far end is ADHD. And that really solidifies the idea that we're seeing these population spectra at the far end being a diagnosis. the other really cool thing is that many people have argued over the years about, is adult ADHD the same thing as pediatric ADHD? And some of these longitude, no, I guess they're not longitude. Some of these studies that looked at adult ADHD and asked, did they also have ADHD in childhood, were really arguing that it was a distinct entity. But, those of us who are child psychiatrists know that people who are very smart can get away with not getting a diagnosis until far later in their lives. People who are female often don't come to the attention of people. So a lot of times you miss that child diagnosis, and in adulthood, if you just look back and ask, did you have a diagnosis? The answer is no, but that doesn't mean it wasn't there, right? And so what they saw when they looked at the genetics between adult ADHD onset and child ADHD onset, they saw they were the same things. So it really does look like one neurodevelopmental disorder. Also, interestingly, the statistics are a little over my head here, but you can use a technique called Mendelian randomization that can look at causal links between things by using the genetics, because those are, those are factors that don't change, and you can see if the same genetics are influencing both things. And so what they saw was there's a causal link between having ADHD and those ADHD variants and having lifetime cannabis use. And then autism genetics. So early on, people used sequencing to identify big, uh, you know, um, very destructive variants that took out genes to identify risk for autism spectrum that were big effects, that were just, you got one thing and it really took you over the edge. But it took us a while to get to the point where we had a GWAS that could look at common variants that predispose to autism. So not these rare things that have big effects, but lots of little things that together push you in that direction. And so what they found was, it was interesting because those common variants aligned well with the big big impact sequencing variants that were identified. So the same sorts of genes came up. So chromatin regulation, the things that like control gene expression by opening the genome or closing the genome off. They found neuronal and synaptic genes that affected development and function. And so they really did converge on the same sorts of things, but just different ways that they were impacted. It also supported the hypothesis that there's a distinct um, genetic architecture between high and low functioning ASD subtypes. So while the low functioning ASDs that are often not heritable in the same sense, where you're not, the parents don't give it to the kids, but there's a new mutation in the kids. In common ASD, these are often families that are very high functioning. And when they did the GWAS between ASD risk genes and IQ, they found that common risk alleles for ASD are associated with higher IQ in the general population. Now, I heard this announced in a room full of geneticists who were surprised by this result. They thought it was going to be low IQ. And I'm sitting there like, really, people? Like, you need to see a patient or two, because, like, I mean, to turn and look at your colleagues, right? Like, I mean, this is like, if you have a little bit of autism, you can do really, really well in life, right? And so this is not at all surprising that it's associated with high IQ to me, and I'm sure to many of you. All right, so I'm going to go into more detail on this one because it's my own data that I did in collaboration with Edie London, my other awesome female mentor at UCLA. And she may be on a plane right now listening to this lecture. So if she is, hey, Edie, hope I represent well. So this just came out on my birthday in August this last year. And we worked a long time on this, so we were very proud that we got it out in molecular psychiatry. And what this study is about is thinking about genetic contributions to risky decision-making, risk-taking in typical individuals and in psychiatric disorders. So the idea here was our hypothesis that given the shift in conceptualizing mental illness from a categorical disease models to these extremes of intersecting population traits, we hypothesize that that genetic variants predicting risk for risky decision making in neurotypical individuals would be enriched in those with mental illnesses, especially those where impulsivity plays a big role, like bipolar, ADHD, and substance abuse. And our aims were to develop and test a polygenic model to estimate the genetic contributions to risky decision making as measured by the balloon analog risk task, which I'll tell you about. And we wanted then to compare our polygenic model of risky decision making to the domain data sets available in order to estimate the genetic risk between risky decision making and psychiatric disorders that are characterized by impulsivity and risk-taking behaviors. So, I told you you can use a PRS to compare traits to each other, to disorders to each other. So here we're comparing a trait, risky decision making, to disorders that we think might have a big piece of that risky decision making as part of their underlying genetics. Oops. So background, risk-taking is a common heritable endophenotype or intermediate phenotype seen across psychiatric disorders. We can measure it with self-report questionnaires or laboratory tests of risk-taking behavior like the Iowa Gambling Task, Cambridge Gambling Task, and the Balloon Analog Risk Test, BART, our chosen task. They have been shown, risk-taking has been shown to be heritable, highly heritable, and relatively developmentally stable. But the underlying genetic architecture is incompletely explored. There was a large GWAS that found numerous associations across several self-reported phenotypes, including drug and alcohol use. The strongest replicating signal was at a neural cell adhesion gene, CAD-M2. And this shared significant genetic risks with schizophrenia, bipolar, ADHD, PTSD, smoking, and obesity. So kind of that link is there as well. And overall, the results suggested that risk-taking is a complex trait that's highly polygenic, many genes, likely driven by many genetic variants, each with a small effect. So we examined risky behavior in the BART in two independent samples of European American ancestry. One was our own UCLA's CNP LA2K sample. This is the Phenomics Center that Bob Builder led way back. And that was over 1,000 samples. And then we also have a colleague, Harriet DeWitt, good friend of Edie's, who formed the genetics of impulsivity sample. And she has just a slightly smaller population. And interestingly, they both did the BART, actually did the task that I'll show you. And so we had genetics on a huge number of people who'd done this task, which really hadn't been done before. And we constructed a polygenic risk score for discovery in each data set and then tested it in the other as a replication. So were we seeing a real genetic effect that was in both independent data sets? We then combined these using a genome-wide mega-analysis, and then we could then use it to test correlation between risky decision-making as measured by the BART and the psychiatric phenotypes that are in the PGC consortium. Especially, we focused on the ones that are characterized by risk-taking, ADHD, bipolar, alcohol use disorder, and prior cannabis use. And we did this a while ago. There's more data there now, but that's what was available at the time. And so the idea on the BART, so that you understand it, is that you are getting points. Sometimes it's money, sometimes it's some other reward. But every time you inflate this virtual balloon, you get a point, but you don't get to keep those points unless you cash out. The more times you pump, the more money when you cash out, but you risk the balloon exploding and then you get no points. Okay, so there's a variable number of pumps for each trial. And so over time, you kind of have to learn what's the right balance between pumping and cashing out in order to do well. And so as you can imagine, people who are very averse to risk taking cash out too early and they don't do well for that reason. And then people that are too risk-taking will blow up the balloon all the time, and they don't do well for that. So we're able to measure how much risk they're taking using the BART on a computer. They're virtually inflating these balloons. So what we saw was that BART performance was confirmed to be genetic, as the same genetic signal was detected in both of our independent data sets. And there's a QQ plot on the right, which geneticists need to see to show that we didn't that we had a good correction, that we don't have any garbage. And you can see that there's one little dot that elevates above the general line. Each SNP is on that horizontal line, and so it's where it's predicted, but then we do have that increase in that one spot, which I'll tell you about. And what we saw was that the PRS that we did for risky decision making based on the BART performance in the GOI in Harriet-Iwitz sample predicted BART performance in the LA2K sample. So it replicated at a very strong p-value even after correction for multiple testing. The FDR is the correction. When the PRS was derived from the LA2K set and then applied to the GIP, so I'm sorry, that's supposed to be GOI. Sorry, she changed the letters on us. So the reverse, start in one, go to the other, we saw the same thing. So it looks real that we're measuring that same risk-taking trait in both populations. That's demonstrated by the BART. And since the two samples are using the same measure of risky decision-making, the BART, we can combine them into one sample. And then we can use that larger sample, it's about 2,000 individuals, to ask questions of the public data. And so this is our GWAS, and here's our Manhattan plot. And we do have this one signal that achieved genome-wide significance. That means it could survive multiple testing for all those, for what we've decided is the appropriate correction for multiple testing of a genome-wide study, 5 to the 10 to the minus 8th. And so we did get one significant hit, which is actually impressive in a sample size this small. You often need like tens of thousands to get a hit, and we got this in 2,000, which is cool. And then this other one is interesting, but not genome-wide significant, but it is significant for the number of actual tests we did. And so we wanted to look at those two genes and see what they were. They both actually make a lot of sense for this kind of disorder. The immunoglobulin superfamily protein IGSF21 is robustly expressed in brain tissue. It's a promoter enhancer that is being detected. It's involved in thalamic and inhibitory synaptic development, interacts with presynaptic neurexin 2 alpha. Knockout mice of the gene have impaired sensory gating, impaired inhibitory synaptic organization, and impaired GABA transmission. And ethanol consumption increases expression. Something's wrong. Ethanol consumption increases expression of the in macaques. So there is these links here to inhibition and substance. The other gene, SRGAP3, is also known as mental disorder associated GAP protein. And it's associated, and the variant that we saw was in the promoter that is predicted to impact promoter function. Rodent studies reveal prominent hippocampal and cortical expression. Gene knockout results in neurodevelopmental, cognitive, and behavioral phenotypes. And a hypothesis role in chromosomal intellectual disability is seen when you lose both genes. When you lose, one gene. And then de novo new missense variants are associated with autism in the Simons collection. And then the last thing we did on this study was we applied the PRS that we had from that combined GWAS to the PGC disorders, and we saw a success in the cannabis use disorder. So we could predict risk for cannabis abuse using the BART gene PRS. So these are shared genetic variants that contribute to risky decision making and cannabis use. And that was again significant and significant with correction. This is me and Edie doing our victory dance after we got that result. Of course, we were a little sad that we didn't get the result in the other disorders. So ADHD, bipolar, and alcohol use disorder were negative, as was a non-psychiatric control phenotype. But in conclusion, we did see that common genetic variants drive risky decision-making measured by the BART, and we saw an overlap with risk for cannabis abuse. What of the other disorders? How come we didn't see association there? Well, certainly given the genetic and phenotypic complexity, we probably needed a much larger sample to do it, to identify genes, more than one at least associated with risky decision-making, and to see an association with these other disorders. And when you look into these public data sets, they're very heterogeneous with limited data, but the cannabis data set was by far the largest, and that may explain the lone positive results. So it may be that as we add more data to the other disorders, we may see the signal emerge. And so we should repeat these analysis with the bigger samples over time as they grow. Okay, moving on to the last section. We are definitely interested in having genetically informed diagnosis so that we can really be able to tell people what they definitively have, what the biology is, what their prognosis is, what kinds of treatments would be good, and maybe even prevention. So this is a huge future goal and dream. Right now, we can do this for disorders resulting from single gene mutation or copy number variants, which is mostly autism spectrum. So you should be doing, if you're a child psychiatrist, and well, even if you're an adult, I think it might make sense, diagnostic genetic testing, which includes a microarray and a fragile X PCR, plus or minus exome sequencing, probably plus as a second if you don't find it with the first. And it's now standard of care for autism, intellectual disability, and developmental delay to reveal causative genetic variants. And you actually get a diagnosis 20 to 50% of the time, which is huge. A diagnosis informs prognosis, associated physical and mental problems. Many of these genes have cardiac issues, have cancer risk, have, various physical endocrine kinds of problems that you could know to look out for. It affects family planning, and it affects eligibility for clinical trials and support groups. So there's value to doing that. But most disorders are genetically complex, and right now we can't do a lot with that. Polygenic risk scores certainly are being done by crazy companies that are telling you your risk for things, but these are not in a place where you get any predictive value for an individual. You really can only look in populations. And so we really can't use those this way. And if you think about it, really, if we wanted to replace the DSM with something that was more accurate to biology, it probably would need to be dimensional. And so would it be something like an Enneagram where you had like, oh, you're a 7 on compulsivity and 6 on internalizing and 4 on psychotic, and you would know kind of a description of what kinds of issues your patient has based on these numbers? I don't know. I challenge you to think about what would be a good way to represent, especially because I think having a diagnosis is valuable to treatment, to patients, to communication, to insurance. And so it's a challenge. Genetically guided treatment is going to be huge, certainly is now in cancer. Oh my gosh, the cancer improvement we've had based on genetics has been phenomenal. And so as we learn more about disorder biology and treatment biology, we'll be able to do a whole lot better than what we do now, which is all, for the most part, trial and error prescribing. But the problem is we currently only know the tip of the iceberg about how drugs work and the genetics that influences the effects. Nevertheless, many companies are offering testing, which are called decision support tools today, based on really sparse evidence. But the bottom line is that current testing is probably a very poor reflection of the complexity of drug effects and of limited value. Okay, here is my audience participation question. Please put in the chat the letter that you agree with. Pharmacogenetic tests currently can tell you which med will work for your patient. Pharmacogenetic tests can help select between indicated options or make dosing adjustments. A patient doing well on a med that a pharmacogenetic test labels red should be switched to a different option. Could be high risk, whatever. Or D, you should not prescribe meds in the red box. Opinions? Anyone? This is a test of how socially anxious people are. Nice. Well, we have enough now that nobody's going to pick something that's not B. If I do this at a conference, I get a lot of answers that are not B. B is the right answer. Oops. And all of these other things are things people have said to me that they're doing. And it's very disturbing when you speak about this to hear what doctors are doing with these tests, and very disturbing to get these test results that lead people to do things that are not in the best interests of patients. So I speak about this a lot. I don't think like you should never use it, but I think you really need to know what you're doing if you're going to use it, because most of the time I see people do bad things with this. So when we talk about drug effects, we're talking about pharmacokinetic effects. So disposition, does it get absorbed? Does it get to the brain? Does it cross the blood-brain barrier? Is it broken down in the liver? Is it transported in the kidney? These things affect the drug levels. And then there's pharmacodynamics, which is the effects at the target. So where it's doing its work. And we know very little about pharmacodynamics of our drugs. Even if we know the initial site it binds, doesn't mean we know where the impact is happening pharmacodynamically. It could be lots downstream, right? And so really the pharmacokinetic variants are the only ones we really should be using right now, with one exception. If you look at psychiatric medications, most of them are metabolized by CYP2D6 and CYP2C19. So these are the ones you really need to worry about. Also because they very commonly have genetic variants that change their function. So if you have a poor metabolizer, that's two bad copies of the enzyme, you're probably going to have more side effects, right? Because you have a lot higher doses, but it might work better. A lot of our drugs are probably underdosed for people who are normal metabolizers. Intermediate, extensive are generally normal. Ultra rapids are a problem because if you have an ultra rapid metabolizer phenotype, which is often an extra copy or an enhancing mutation, then you're going to have lower bioavailability and possibly efficacy. You may never get to therapeutic levels. So these are things you should think about if you have the genetics anyway. Whether you should order the genetics is another case, but if you have them, why not look at them? So very common, you're treating people with these things all the time. So you're usually adjusting for it by, seeing what's happening. But if you know up front, maybe you can do better. A lot of people are convinced atomoxetine doesn't work. Strattera, it's a second line medication for ADHD, not a stimulant. Well, the thing is, genetic variation at CYP2D6 predicts outcome with atomoxetine use. So the variability in the drug levels is 10 to 30 times between the normal and poor metabolizer. That's a huge difference, right? Therefore, the poor metabolizers get more benefit and more side effects. And in fact, the FDA really made the dosing guidelines so that it would be safe for poor metabolizers. But what it means is it's inadequate for normal metabolizers. So a third of individuals require doses above the FDA limit to achieve a therapeutic level. So the reason we all think it doesn't work is because we never dose people appropriately. So really, the CPIC paper that I'm part of really argues for going above the FDA limit if they're not having side effects, because they could be a normal metabolizer, and we could really do better. We also recommend doing drug levels if you want to dose it appropriately. The decision support tools, when we look at those, what are they testing? So they do include these pharmacokinetic variants that can be useful, but they also include a bunch of pharmacokinetic stuff that's not useful. And they also include a bunch of pharmacodynamic stuff that is based on really poor, weak data, some of which I participated in generating. That is, back in the candidate gene days when people were having all kinds of associations that didn't hold up. So the idea that you'd be making clinical decisions based on, bad data is disturbing. So anytime you see serotonin, adrenergic or dopaminergic anything, ignore it. It's garbage. If the serotonin transporter was real, it would have showed up in our GWAS by now. It hasn't. Okay. Test interpretation. So I hate these stoplight colored things. Even ones that don't do it still label it as like caution advised, or avoid, right? And so, but the problem is just because it's in the green or good bin doesn't mean it's going to work. It just be less likely to cause side effects, maybe. And really, most people interpret the green as this is the drug that's going to work for your patient, but you know, that's not it at all, right? And you guys know that, which is great. So current testing doesn't tell you which drug to pick, only which you might want to avoid or dose differently. There is a meta-analysis. There's actually more than one meta-analysis, but this one is this is a good one. It's by someone I think is quite good. But you know what? If you put together a bunch of crap data, you get crap meta-analysis. Um, anyway, most of the data supporting these things is open label with no randomization or blinding. And if you tell someone this is, you know, this genetic test is going to tell us which drug to do, you get a huge placebo effect. So open label is always going to be positive. Many of the RCTs don't achieve their primary outcome. They use their secondary outcomes and they're often post hoc. They don't correct for multiple testing. They're really not valid. And they're all commercially sponsored until recently, which have a lot of bias and conflicts of interest. And the ones that haven't been commercially sponsored have been negative. The population they use is limited, generally middle-aged women who have failed at least one prior trial. So when we're using it in child, we're extrapolating from middle-aged women with a totally different diagnosis, probably. Oh, I should have stuck in there. Depression. It's always depression that they're testing. And there's issues with blinding. There's issues with publication bias. Who's going to publish a trial that's negative if you're trying to sell the test? And so if you look at the meta-analysis, you get a positive, but I just told you why these studies all have positives and it's not really legit. Additionally, the meta-analysis factors don't look good either. So I don't think there's a lot to take from this data. And there was nothing in children until 2021. And when the first child one emerged, you know what, it was totally negative. So they looked at depression in teens. They compared a GeneSight report with treatment as usual, and they saw nothing at 8 weeks or 6 months for symptom improvement, side effect burden, or satisfaction. And MDs and the gene arm, though, they prescribed less of the appropriate drug because SSRIs are the only thing that has evidence. And yet they were doing something else. And that was their only significant finding, which didn't surprise me one bit because I see it all the time. So why do we see this? You're supposed to prescribe SSRIs. The treatment as usual do that. They might do a little bit of the other things for some reason. But the GeneSight people are doing like some wacky stuff here. If we were in person, you would tell me why it happens, but it happens because all of these new fancy things that have never been tested in children, and some of which have negative data in children, like Prestige or 40-oxetine, these things always land in the green box. And so people always put their patients on these just because they're green, but that doesn't mean they work in the disorder. People are always like, okay, so why are the most expensive meds always in the green box? Well, it's not collusion as much as I think these things are, you know, companies that this is because things are made to have less drug interactions. So these new things always end up in the green box. But this makes docs prescribe off-label and they choose meds without indication or evidence based in a clinical situation. So my take-home message is please always choose the evidence base over the green box. Current evidence-based testing, CYP2D6 and CYP2C19 are reasonable to consider. The other ones are not ready for clinical use. And then the only pharmacodynamic thing is the HLA B1502 that predicts the Stevens-Johnson and other cutaneous side effects from carbamazepine or oxcarbazepine, but importantly, not lamotrigine. A lot of people don't know that. So really, you're just going to use your usual skills, and then you're going to use the genetic data only to think about avoiding a terrible side effect or titration planning or choosing between medications in the same class that have an indication or evidence for the target disorder. I do think it can be useful in a patient who's having a lot of failed trials or a lot of side effects because it's reassuring for the patient. A lot of times I treat anxiety, so these people have a lot of anxiety about medication. So knowing that this thing is unlikely to give you side effects or likely to be a good drug can actually be a useful, powerful thing that can help them stay on the medication. To have better DSTs, we're really going to have to turn to what worked for genetics. We're going to have to do big data approaches, which is hard to do with drug treatment because it's a really expensive study. And we're also going to have to incorporate non-genetic information that is important in drug effects. The cancer people do this. These data sets don't exist right now, but maybe as we start having more electronic health records, we'll be able to leverage that more to be able to move things forward. So just in summary, I'll try to do this fast so you can ask a question or two. Psychiatric disorders are genetic but complex. We really had to scale up to start having victory. Recent efforts have found some really cool stuff, and they really emphasize overlap between disorders, how correlated things are. In practice, we are seeing just the beginning of molecular diagnosis, like in autism, but this will expand over time. And how is it going to change psychiatric disorder classification? We have genetically guided treatment right now, but it's pretty bad. So we need to learn from past lessons to move forward and do a better job with genetically guided treatment. And this is just interesting. I didn't know this, but there were 8% of the genome was hidden until last year. So they just filled in the gaps, some of those areas that were harder to reach. So we're going to keep learning more and more about genomics, and we're not anywhere close to knowing all of that now. This is my last comment. If we knew who was at risk, we have a lot of tools we could use for prevention. Whatever your favorite disorder is, you can think about the things you would do, and you know better than me, to be able to prevent. Like if you told the parents when they had a baby, your child's going to be at risk for anxiety, we would do a lot of behavioral stuff early on to make sure that that didn't happen, that they could manage it. And I think that is true in other areas as well. I'm going to take questions while I leave this slide up because it's entertaining and very Gattaca.
Thank you so much, Dr. Nurmi. This was, wow, a lot of information. We've got a bunch of questions, but the majority of them centered on cannabis and about whether what you were doing, whether the correlation was with cannabis abuse or use, and whether or not you thought that that was really a reflection that cannabis is or was illegal in many places. So it would seem like maybe that would make sense that it would correlate with high risk taking.
Yeah, they're interesting questions. It's complicated because, so I can't just be about the mother using cannabis because the link is with the genetics. Now I guess you could say the mother shares some genetics, but I'm going to get in the loop here. But the link is not with like exposure to cannabis. The link is that they have that genetic variant, and that leads down the road to cannabis use. So I don't think the mother thing makes sense unless you start invoking that the mother shares genes, and then that's just super complicated, and I don't know how you take it apart. Sometimes I said cannabis use, and I'm sorry, Spar, I don't know where I said what, but the study we did was about, it was a cannabis abuse group, but they were actually, the question that they were using was about use. So again, that's complicated, but that was what they asked. We didn't have any control over that. Um, so huh.
Yeah, and then that was about risk taking. I mean, we're at 11:59, so you can choose one more of those that you want to answer, or there was also one question about whether or not you would want to do genetic testing for lower than expected plausible levels.
Oh God. Okay. Well, I'm actually super interested in the first question because it's an OCD question. But I think like, you know what, SSRIs don't work all that well for OCD if you don't have therapy as well. And so I think what SSRIs do really well is enable the person to do the exposure therapy on its own. It's like not great. And so so I would suggest that for anorexia, we need to pair the behavioral interventions with the SSRIs, which I think we're certainly doing in the areas we've made some progress. But the overlap is, you know, some genetic underlying issues, but then you have a whole bunch of other things too. So it's super complex. So I don't know that we could ever say the same meds. Like, I mean, OCD and Tourette are very linked, but the Tourette meds are different than the OCD. So I think it's much more complicated than we think. The I can answer this real quick. The MFR deficiency testing is weak data. It's only really been seen in predicting treatment to depression, and only if you have two variants, but both chromosomes, not one, because having one is super common. Sorry, I didn't get to
Yeah.
all the
No, that's fine. And actually the only problem is that the audience can't see the questions. So I guess
Oh,
it was obvious to people that what Dr. Nurmi was responding to was a question about why would SSRIs then not work for anorexia nervosa, given the overlap with OCD. And the answer is it doesn't really work very well for either of them. It's the psychotherapy that makes the difference. So this is wonderful. We do want to have you come back and tell us the new developments.
thanks so much.
Thank you. Okay. Bye-bye.